Anti-CRISPR systems are a form of biotechnology associated with genome editing, specifically the CRISPR-Cas9 system. Anti-CRISPR's technology's purpose is to interact with specific components of CRISPR-Cas9 systems, such as effector nucleases, to avoid the destruction of phage DNA. The anti-CRISPRs are used to control genome editing strategies to reduce unwanted outcomes and potentially improve the safety of the genetic editing technology. So some limitations are seen with the anti-CRISPR technology. The CRISPR-Cas9 system can sometimes cause unintended sequence alterations at non-target sites, which are referred to as off-target events. And these are typically caused by mutations or degradation. There are some other examples of genetic engineering technology, such as the Cas-clover nucleases, CRISPR CPF1, transcription activator-like effector nucleases, or TALENs. So the Cas-clover nucleases are going to utilize two guide RNAs, while the CRISPR CPF1 is going to utilize one RNA, and the transcription activator-like effector nucleases are going to use restriction enzymes in their processes. So an advantage of using CRISPR technology over the aforementioned technologies is CRISPR-Cas9 is faster, cheaper, more accurate, more efficient than other genome editing models that are available. A CRISPR-positive control is needed to monitor the gene editing efficiency of the CRISPR reagents that are being used in the experimental design. The figure above is from the paper Anti-CRISPR Protein Applications, a natural break for CRISPR-Cas9 technologies, and it depicts the stages of CRISPR-Cas9 immunity during adaptation, a fragmentation of invading nucleic acids while it's incorporated as a new spacer in the CRISPR array. It depicts the biogenesis stage where the CRISPR array is transcribed into a long precursor CRISPR RNA that is then processed by the Cas proteins into a single CRRNA that acts as a guide for the Cas effector nucleases. This figure is derived from the paper Anti-CRISPRs, the natural inhibitor for CRISPR-Cas9 systems. It shows the schematics of anti-CRISPR protein interference with different stages of type 1, type 2, and type 4 CRISPR-Cas systems. And if you look closer at the diagram, you can see in part B here where the ARC is able to interfere with the binding, and you can see the difference between the two diagrams where the ARC is not present in diagram A versus diagram B, and how that affects the binding that is able to occur. These figures are derived from the paper Anti-CRISPR ARC2C5 is a dsDNA mimic that inhibits type 2C Cas9 effectors by blocking the PAM recognition. You can see the ARC2C5 binds this complex to inhibit the dsDNA cleavage, and these figures are able to determine through the assays quantify the differences between these recognition sites.